Okay, students, good morning, all of you. Our next topic discussion cloning sites. Cloning sites that is already we have kept a discussion. Next, so the example and technique of selection of recombinase by inactivation of one antibiotic resistant gene is a cumbersome process. Cumbersome and drawn long drawn process. Cumbersome and long drawn process. The example and technique of selection of recombinase. Selection of recombinase. Selection of recombinase. Selection of recombinant by inactivation of one antibiotic gene is a cumbersome. Cumbersome means difficult. Cumbersome, difficult, and it takes a long time. Long time and extremely difficult. See here, yesterday I said that due to insertion of foreign DNA, the tetracycline gene is lost resistance, but still remains ampicillin resistance. Okay, so when recombinant DNA is transferred into the bacteria grown on ampicillin containing medium, but not tetracycline medium. So the bacteria which are not grown on tetracycline medium, those bacteria are called recombinant bacteria or transformant bacteria. Okay, so the example and technique of selection of selection of recombinants, recombined DNA by inactivation of one antibiotic gene is a difficult and much time taking process. And also it requires antibiotic containing culture plates. Antibiotic containing antibiotic containing culture plates. Antibiotic containing culture plates are required. One or two antibiotic containing culture plates. So antibiotic containing culture plates means what? Artificially some plates we have to prepare. How artificial DNA and RNA are synthesized like that. So artificially antibiotic containing plates they have to prepare and used in cloning sites where foreign DNA is inserted. So like that, it is a long drawn, it is a difficult process. Antibiotic containing culture plates preparing, it's very difficult process in biotechnological applications. So hence, alternative method scientists are developed. Alternative method scientists are developed. So that alternative method now we will see. Okay, it is a difficult and a long time taking process. So that much time spending, it's very difficult task. Okay, so in such cases, alternative method, there is alternative method they use. Nothing but the alternative selectable markers. Alternative selectable markers they use. See here, alternative method is the use of selectable markers. Alternative method means what? In that method, they will use selectable markers. Alternative method is the use of selectable markers. Alternative method is the use of selectable markers, which helps in. Alternative method is the use of selectable markers, which helps in identifying recombinants from the non-recombinants on the basis of producing the color from the chromogenic substrate. From the chromogenic substrate X-galactoside. Okay, so come to the point. Alternative method is the use of selectable markers. Use of selectable markers. These selectable markers helps in identifying, helps in differentiation. These selectable markers helps in identifying or differentiation from recombinants. From recombinants to non-recombinants. So selectable alternative method means use of selectable markers that helps to differentiate between the recombinant from so those selectable markers helps in differentiation between recombinant from non-recombinant on the basis of their ability to produce color with chromogenic substrate. Chromogenic substrate X-galactoside. 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 That's all. Okay. So once again, this first point I am saying isn't your alternative method. So in this alternative method is the use of selectable markers. These selectable markers helps to differentiate between recombinant and non-recombinant on the basis of recombinant and non-recombinant ability. On the basis of this ability to produce color with chromogenic substrate, that is X-galactoside. Okay, so recombinant and non-recombinant DNA, recombinant DNA and non-recombinant DNA have the ability to produce color with the chromogenic substrate. On the basis of their ability to produce color, you can differentiate which is recombinant, which is non-recombinant. Okay, so that is the first point. Once again, the first point. Alternative method is the use of selectable markers. Selectable markers means genes only. Those are also genes only. So those selectable markers helps to differentiate between the recombinant and non-recombinant on the basis of these. On the basis of their ability to produce color, indigo blue color with chromogenic substrate X-galactoside. In our textbook, X-gal they given. Actually here X-gal, not X-galactoside. X-galactoside. So here in alternative method, if the vector is used PUC18, if the vector is used PUC18 in yesterday cloning site is used TR322, T plasmid, B Bolivar or Rodriguez. Those scientists restructured that. Artificially restructured. Like that only one more vector. P stands for plasmid. U stands university. C stands for California. University California. The numerical designation 18. Why means number of PUC vectors are there. 19101 is there. 19102 is there. Like that. Number of artificially restructured vectors are there. To differentiate one vector to another vector, they have given some simple numerical designation. So PUC vector 18 used for alternative method. In this one, lack that gene in the PUC 18 vector. It is also same, the PUC vector. So it is also the vector same like this only we did. Same, like this only, that is vector, plasmid vector, like that. So, in this vector, lac Z gene, it can synthesize this. Beta galactosidase enzyme. LACZ gene can synthesize this beta galactosidase enzyme. So this beta galactosidase enzyme will react with chromogenic substrate, chromogenic substrate, chromogenic substrate X galactoside. Okay, so LACZ gene synthesizes beta galactosidase which react with the chromogenic substrate, that is X galactoside, and convert into galactose 4-chloro bromo compound. Galactose and 4-chloro-3-bromo compound, which later gradually converted into indigo blue colored compound. Indigo blue colored compound. Indigo blue colored compound. What I said that alternative method is the use of selectable markers. Those selectable markers helps to differentiate between recombinants and non-recombinant on the basis of this ability to produce color with chromogenic substrate. Chromogenic substrate X-galactoside. On the basis of their ability to produce color, what color? Indigo blue color. On the basis of their ability to produce color with the chromogenic substrate X-galactoside. X-galactoside alternative method, selectable markers. Selectable marker, nothing but G only, that is LACZ gene. It is reacted with X-galactoside and convert X-galactoside into indigo blue color. So this process can be performed by non-recombinant DNA. That means not alternative DNA. Non-recombinant DNA means any alternation does not happen. That DNA only can be color indigo blue. Okay, that is one statement. Next statement here. If alien piece of DNA is introduced, this is PUC18 method. PUC18 method. If alien piece of DNA, alien not foreign DNA. If alien piece of DNA, nothing but alien piece of DNA. Alien piece of DNA. If alien piece of DNA is introduced in this region, so same process. If alien piece of DNA is introduced at the LACZ gene, here actually originally this is PUC18 method. Here LACZ gene is there. In this region, they cut to introduce alien DNA. They cut in this region. After cutting this region, it would be appear like this. Here, what gene is there? LACZ gene. The LACZ gene they cut. They introduce foreign DNA, alien piece of DNA. So when alien piece of DNA is introduced at the LACZ gene, then it is called recombinant DNA. So this recombinant DNA does not produce beta galactosidase. Why? The LACZ gene, here LACZ gene, the LACZ gene was not intact. The LACZ gene was not intact because it was cut. It was cut off for insertion of alien piece of DNA. Then LACZ gene was cut off. Foreign DNA is introduced. So due to the absence of LACZ gene, it will not synthesize beta galactosidase enzyme. If the beta galactosidase enzyme is not available, this recombinant, when it is introduced here, it is called recombinant DNA. It is called recombinant DNA. This recombinant DNA will not produce color, will not react with X-galactoside, because in that recombinant DNA, beta-galactosidase enzyme is not there. Beta-galactosidase enzyme is not there. Therefore, recombinant DNA is not reacting with X-galactoside and it will not produce indigo blue color. Indigo blue color, because the LAC-Z gene was inactivated. The LAC-Z gene was inactivated due to the insertion of foreign DNA, foreign gene, desired gene of our interest. When foreign DNA is inserted at the PUC18 at cloning site, at specific site, that is LAC-Z gene. So the LAC-Z gene was not present. So for insertion of foreign DNA, LAC-Z gene was cut off. Due to that cut off, the LAC gene was inactivated. It was not complete. LAC gene was became half, so that it was not activated. Okay, so then it's called recombinant DNA. This recombinant DNA is not reacted with chromosome substrate X-galactoside and does not produce indigo blue color. What I said in that first statement, alternate method is the use of selectable markers, which help to differentiate between the recombinant and non-recombinant on the basis of their ability, his ability to produce color with chromosome substrate X-galactoside. If color is produced, that is non-recombinant. That DNA is not, that vector is not modified, non-recombinant. If color is not produced, that is called recombinant. Okay, so the bacterial cells, the bacterial clones which don't produce color, those are called recombinant types. Those are called recombinants. Okay, so this is one statement. So these bacterial cells, see, foreign DNA is inserted. Then this is transferred into the host cell, E. coli bacteria. E. coli bacteria. So E. coli bacteria are transferred into the medium containing chromosome substrate X-galactoside. That bacterial clones could not produce indigo blue color due to the insertion of foreign gene. Due to the insertion of foreign gene. Due to insertion of foreign gene. It is called inactivation of gene due to insertion. Hence, the bacterial clones called recombinants. Bacterial clones are called recombinants. The bacteria could not produce color with chromosomic substrate. Therefore, those bacterial clones, those bacterial cells are called recombinants. On that day, selectable markers, when I am saying, I said, in addition to the ORI, cloning vectors requires selectable markers. Those selectable markers helps to identify and eliminate non-recombinants and selectively permitting the growth of recombinants. Discuss that means what selectable markers are gene only. In the case of PBR322, ampicillin, tetracycline, these are selectable markers. These two, one is inactivated due to insertion of foreign DNA. One is activated. Okay, so the gene which is inactivated, that is called, that will support recombinants. The gene which is inactivated, that will support recombinants. That is tetracycline gene supports recombinants. Okay, so this is called cloning sites. Only one point you have to remember here. So due to the insertion of foreign DNA, for insertion of foreign DNA, existing gene we are cutting off. We are cut off. Due to cut off, the naturality of that particular gene is diverted, converted. Then that particular gene gets inactivated. If that gene is inactivated, automatically the gene product also inactivated. Okay, so this is called alternative method. So selectable markers means what? Genes only. Okay, in one vector, one type of a genes acts as a selectable markers. Okay, so here one mark definition. What is that definition? So what is insertional inactivation? What is insertional inactivation? When foreign DNA is inserted into the one of the antibiotic resistant genes, one gene gets deactivated. That process is called insertional inactivation. Due to insertion of foreign DNA, one gene gets inactivated. Automatically, that gene product also suppresses. That process is called insertional inactivation. Due to insertion, the gene is inactivated. Due to insertion of foreign DNA, that particular gene is inactivated. This phenomenon is called insertional inactivation. Okay, so the colonies which cannot produce color, those colonies are recombinants, recombined. Okay, so that's it. So with this cloning sites, okay, the features, some features are required to facilitate cloning into vector. Those features are successfully finished. Okay. Okay. Competent host. Competent host. Key. Tools of recombinant DNA technology. That I said that. First tool is enzymes. About enzymes, extensively we discussed. Second tool is cloning vectors. About cloning vectors also successfully, extensively we discussed. Third one is competent host. So the DNA is hydrophilic molecule. DNA is hydrophilic molecule. Hydro means water, philic means attached. Hydrophobic. Phobic means fear. Hydrophobic, water fear. Hydrophilic, water attractive. Okay, so the DNA is a water attractive molecule. The DNA is a hydrophilic molecule and cannot pass so easily into the host organisms. We discussed that recombinant DNA is transferred into the E. coli bacteria. We discussed earlier. So the recombinant DNA directly cannot pass into the E. coli cells. It's not so easy. It's very difficult process. Okay. So the DNA is a hydrophilic molecule and cannot pass easily into the host cells. First, the bacteria must first be made competent to take up the DNA. First, what we have to do? The bacteria must ready to take the formula. In such a way, we have to make the bacteria. Okay, so that is the first statement. DNA is a hydrophilic molecule and cannot pass the DNA directly into the host cells. Okay, first must the bacteria made ready to take up DNA. For that, methods we have to follow. So the transfer of foreign DNA into the E. coli bacteria is called gene transfer. The transfer of foreign DNA into the E. coli bacteria is called gene transfer. The gene transfer methods are two types. Gene transfer techniques are two types. One is indirect method. One is indirect method. Another one is direct method. Indirect and direct methods. Okay, so the transfer of foreign DNA into the E. coli bacteria can be performed by two methods. One is indirect method, another one is direct. So first of all, let us see indirect method. Indirect method. Indirect method, nothing but vector method. Vector method. Already we have seen that. We will use the different kinds of vectors, that is plasmids, agrobacterium tumefaciens, retroviruses, bacteriophages, cosmids, artificial chromosomes of bacteria, yeast, plant and animal viruses, jumping genes or mobile genes. These are all used as vectors for transferring foreign DNA to the host bacteria. That is called indirect method. So that's already we have discussed that. Agrobacterium tumefaciens. It is a pathogenic bacteria for dicotyledonous plants, especially for dicotyledonous plants. Agrobacterium tumefaciens is a pathogenic, causes disease. Okay, so that kind of the agrobacterium tumefaciens is corrected and disarmed. Pathogenic nature is converted into natural, neutral. Those agrobacterial tumefaciens are used as vectors. That is already we discussed. Already I said that. In the same way, retroviruses. It is also causes normal cells into cancerous cells. Okay, so the retroviruses are disarmed. Pathogenic nature is neutralized and used as a vectors. That is called indirect method. To transfer foreign DNA into the host bacteria, use vectors. There are number of vectors used to transfer foreign DNA. Okay, that is indirect method. Next, direct method. So direct method nothing but vectorless method. Direct method nothing but vectorless method. Without any vector, directly foreign DNA is inserted into the E. coli bacteria host cell. So indirect method. So this can be performed by three different types of techniques. One technique, first one is electroporation. One is electroporation. Second one is microinjection. Second one is microinjection. Third one is biolistic or gene method. Okay, so this is I'm like earlier I am planning to write here. Actually, it is the second EMB electroporation, microinjection, biolistic or gene gun or gene particle gun method. Electroporation. So in this technique, host bacteria are temporarily applied with electrical impulses. See, this is E. coli bacteria. This is E. coli bacteria. So these E. coli bacteria are temporarily treated with electrical impulses. Electrical impulses. Electrical impulses. Electrical impulses. So electrical impulses are exposed. So those electrical impulses makes holes on the plasma membrane of the bacteria. Those electrical impulses makes holes on the plasma membrane of the host cells. Then we can insert through these holes foreign DNA into the coli bacteria. This is one thing. Before applying electrical impulses, so these bacterial outer cell wall is chemicals, that is calcium chloride. Calcium chloride. So this bacterial cell wall is treated with divalent chemicals, that is calcium chloride. Okay, when they are treated with divalent calcium chloride, after they will apply temporary electrical impulses. Those impulses keeps holes in that plasma membrane of the bacteria. Through that holes, foreign DNA is directly injected into the bacteria. In the bacteria, what happens? Everybody knows that multiple copies are produced with the help of DNA polymerase enzyme. Those multiple copies are transferred to our interested plants or animals. So this is one in electroporation. In this only heat shock treatment. Heat shock treatment. In this only one sub. So foreign DNA and bacterial cells. The bacteria, foreign DNA and the bacteria are incubated on ice. First incubated on ice, followed by a brief exposure to heat about 42 degrees centigrade. First they will be incubated on ice. After that, they will be exposed to heat shock treatment about 42 degrees centigrade. After heat shock treatment, again they will be incubated on ice. Okay, bacteria and foreign DNA. Bacteria and foreign DNA is first incubated on ice. After they will be applied heat shock treatment. Briefly they will be exposed to heat about 42 degrees centigrade. After finishing that, again they will be incubated on ice. Then foreign DNA is inserted into the bacteria. Next, micro injection. So foreign DNA is directly inserted, directly injected. Foreign DNA is directly injected into the nucleus of plant or animal through micropipette. Micropipette. So micropipette they will take. In that micropipette, they will take foreign DNA. So that foreign DNA directly injected into the plant or animal cells. Directly they will be injected into the plant or animals. Biolistic or gene particle gun method. In this one, microparticles, small particles of gold or tungsten is treated with foreign DNA. Gold or tungsten particles they will take, they will be applied on foreign DNA. After applying gold or tungsten on foreign DNA, with high velocity, high pressure, they will be bombarded directly onto the E. coli bacteria. Okay, so that is called gene particle gun method. Gene particle gun method or biolistic means what? In this one machine they will use. In that machine, gold or tungsten particles are applied with foreign DNA. With high velocity, they will be bombarded directly on E. coli bacteria. Then foreign DNA is directly entered into the E. coli bacteria. Foreign DNA is directly entered into the E. coli bacteria. So this is called direct method, vectorless method. Okay, without using vector, directly they can transfer into the E. coli bacteria or host bacteria. This is called gene transfer. For these two methods, one is indirect method, vector method. Another one is direct method, vectorless. Okay, so with this, today's session is finished. Tomorrow we will see process of recombinant DNA technology. With the process of recombinant DNA technology, our biotechnology topic also finished. So today meeting session is finished. You can leave the meeting.